Okay, good morning class. So this is the continuation of your discussion video for the chapter 6, okay, about enzymes. Okay, so we are going to start on the topic industrial strength of enzymes. Okay, so from this point class, from this one, okay, so kung napansin nyo sa baba, wala na tayong mga solving, wala na tayong mga writing structures. These are all concepts, okay? So, mostly explanation lang yun dito. Yung iba naman is self-explanatory na. So, we will just go over the topics or I will just mention additional informations if meron. Okay? But mostly these are just topics. Okay. So, industrial strength of enzymes. Okay, so your enzymes are also used. It is widely used in many industrial or in many types of industries such as your food processing, okay, sa mga pagkain, in technology, in consumer goods such as your detergents, then medical uses also, ginagamit siya in producing pharmaceuticals, okay? So let's first start, okay? So enzymes are not only important in biology but also in the industry, okay? So when you say industries, these are producing, okay? The production of different types of goods. Okay, so there are two major advantages of using enzymes to manufacturing processes and in commercial products. Okay, so first, okay, so ito yung mga advantages when you use enzymes in producing commercial products. Okay, so usually sa factories ito. Okay, first, there is a very large increase in reaction rates at room temperature. So, syempre, diba, the main function of your enzymes is to increase the rate of reaction. So, when you use enzymes in the production of commercial products, okay, or in factories, diba, mas mapapabilis niya yung production ng goods kasi it increases the rate of reaction. So, hindi mo na kailangang i-change yung temperature because kahit na room temperature lang yung ginagamit sa factory, you can still make the production of your products or goods very fast. Secondly, they are relatively specific and can be used to target selected reactants. Okay, so in factories kasi you use many types of reactants, okay? So you by using enzymes, okay, very specific sila ng tina-target nila na reactant. So you will have no trouble na. Wala ka nang poproblemahin sa pag-proceed na reaction in your factory because you are sure that the enzyme that you use will only target a specific reactant in order for a specific reaction to happen. So, meron din siyang disadvantages, okay? So, syempre, since your enzymes are made inside a living organism, mahal yung production niya, di ba? So, ang biggest disadvantages mo if you are using enzymes in factories is that it is scarce, okay? Scarcity and higher cost compared with traditional processes. However, developments in biotechnology offer less expensive enzymes using genetic engineering. So, bakit scarce? Because, syempre, hindi ganun karami yung mga nagpo-produce ng enzymes, okay? Because your enzymes are produced inside a living organism. Okay, so pwede rin naman siyang artificial but it takes long and it takes a lot of money. So, kailangan sa living organism mo siya kinuha. So, hindi siya ganun karami sa market. And higher cost, so yung production niya is medyo mahal. But now, using biotechnology, so when you say biotechnology again, this is the use of microorganisms to produce certain products. Okay, so by using biotechnology, we can produce less expensive enzymes. Okay, mas mura na yung paggawa sa kanya by using genetic engineering. Okay. So many of the common industrial enzymes have a digestive or breakdown function. Okay. So when you say digestive or breakdown function, it breaks down your polymers into monomers. Okay. So usually sa industries natin, they use raw materials. When you say raw materials, ito pa yung untouched types of materials. So, usually, yung raw materials ninyo are made of polymers, okay? Such as your cornstarch, di ba? Malalakit silang type ng molecule. So, in industries, usually, ang ginagamit na enzymes are the ones that has the action of digesting or breaking down polymers to monomers. So, usually, they are hydrolases. So, when you say hydrolase, is the use of the water molecule to break or separate linkages or bonds. So, ito yung binigay niya na example. You have proteases or these are proteases. Pwede mo rin silang tinatawag na peptidases, okay, or proteolytic enzymes. Okay, ito yung specific name nila. So, these are enzymes that break down your proteins. Okay? So, ito yung mga general names for your different or the general names of the enzymes that break down your different biomolecules. So, for lipids, you have lipases, okay? And for your carbohydrates, meron kang apat. You have the cellulases for the breakdown of cellulose, amylases for the breakdown of your amylose, lactase or lactases for the breakdown of your lactose, and finally, pectinases for the breakdown of your pectin. Okay, so let's go to food processing. It used the enzymes for a long time. Okay, so kahit noong panahon pa, ginagamit na yung enzymes for producing food. Okay, so the citrus industry, when you say citrus, ito na yung mga oranges, okay, the family of citrus, okay, use the enzyme pectinase to remove the peel from oranges or grapefruits using vacuum infusion process. The pectinase penetrates the peel. It dissolves the albedo, or the white stringy material that attaches the peel to the fruit. Okay, the skin can be peeled easily by machine or hand when the fruit is removed from the solution. They market the pre-peeled citrus to hospitals, airlines, and restaurants. So, dito, di ba, napansin nyo kapag nag-open kayo ng fresh na orange, mahirap tanggalin yung kanyang outer covering. Because, meron siyang tinatawag na albedo, yung white stringy material. So, I know that you've already seen this materials, lalong-lalo na sa oranges. So, one enzyme that is used to remove this albedo is your pectinase. Okay? So, syempre kapag sa industry ka, sa food processing, dapat mabilisan na, di ba, yung paggawa ng food mo. And kapag inisa-isa mo pa yan, pag peel ng orange, napakatagal. So, yan. Para mas mabilis yung process, ginagamit yung enzyme na pectinase. So, they market yan. So, usually sa hospitals, okay, para sa pagkain ng mga patients, for the airline. So, prepared na, no, wala nang peeling process doon, and restaurants. So, on industrial scale, the enzyme lactase is used to convert lactose to glucose and galactose. The result allows the lactose-intolerant people the ability to consume milk products. Okay? So, you can actually use your lactase in producing like ice creams and other milk products in a way that the lactose intolerant people will be able to eat them. Okay, so your lactase reacts only with lactose, and the glucose and galactose formed are sweeter than the lactose and differ in solubility. Okay, so yung napoproduce na product ng lactase, which is the glucose and the galactose, are sweeter compared to your lactose. So, 'di ba, ang glucose natin and galactose, they are consumable by your lactose intolerant people. Okay, so i-convert ng lactase in lactose to glucose and galactose so that it can be consumed by these types of patients. Okay, so the ability or the products have different properties than lactose. So sabi nga mas matamis sila and they differ in solubility in water. So lactase treated milk is used for ice cream are more creamier. Yeah. So cornstarch, okay, you also have your cornstarch that is used for the food processing industry. So cornstarch are also converted into syrup using industrial enzymes, okay, that are equivalent in sweetness and in calories to ordinary table sugars. Okay. So your cornstarch is not hindi siya directly nako-consume. You will need to convert it to a certain type of syrup before it is edible to human. So kapag na-convert siya into syrup, equal lang yung sweetness niya and yung calorie content niya sa table sugar ninyo, which is the sucrose. So annually or every year, more than 10 million metric tons of this syrup are produced. There are 3 enzymes used in the process such as, okay, so may 3 kang ginagamit na enzymes in converting your cornstarch into edible syrup. You have your alpha amylase. So the first step is to use alpha amylase to catalyze the liquefaction of your starch to dextrine. Okay, so from being a starch or cornstarch, you will convert it to dextrine using your alpha amylase. Ano ba yung dextrine? So your dextrine is actually a low molecular weight carbohydrate. Okay, so it is produced by the hydrolysis of your starch or glycogen. So pwedeng dalawa ang origin ng dextrine mo. Pwedeng from starch, galing sa plants, and pwedeng from glycogen, galing sa animals. So in our case here, cornstarch is from plants, okay? So the second type of enzyme is the glucoamylase, okay? This catalyzes the breakdown of dextrins to glucose. So galing sa dextrine, that is a product of your alpha amylase, your glucoamylase, okay, it converts or it catalyzes the breakdown of your dextrins to glucose. And finally, you have your glucose isomerase that converts glucose to fructose. So, mas matamis yung fructose natin compared to your cornstarch. So, the final product is a high fructose syrup, okay, which is very sweet. That is equivalent in sweetness with your sucrose or table sugar. So, since 1968, yan, tagal-tagal na, one of these syrups are commercially sold containing dry weight about 42% fructose, 50% glucose, and 80% other carbohydrates. So, diba, most of its components are very edible or consumable by your normal people. Okay, so technology. Let's go to technology. Okay, your enzymes are also used in, or this is the use of industrial enzymes. Okay, gave solutions to some industries in their environmental pollution problem. So when you say industrial enzymes, ito na yung enzymes that are produced commercially and is used commercially. Okay, ginagamit siya usually or nabibili na siya sa market, itong types ng enzymes na ito. Okay, industrial enzymes. Okay, so traditionally cotton was scored with a strong base, sodium hydroxide or NaOH. Okay, so gumagamit ka ng strong base para malinisan yung cotton mo, okay? Your fresh cotton or freshly harvested cotton is scored Or when you say score, this is cleaning or brightening of the cotton. Okay. Using a strong base to prepare it for dyeing. However, wastewater from this process is harmful to the environment. Bakit nga? Kasi gumamit ka ng napakalakas na base solution. Okay? So, hindi siya maganda kapag napunta siya sa soil. Okay? So, an enzyme pectinase has replaced successfully this toxic chemical, which operates under milder conditions and achieves the same scored result. So, mas healthy sa environment itong pectinase, the use of pectinase. Because it is natural, okay? Galing din siya sa organic or sa living organism. Okay. Cellulase is also used by a number of industries. So it is used by denim manufacturers to give a soft appearance to the cotton fabric, stonewashing, or biostoning the cloth. Okay, so when you say stonewashing, this is also known as stonewashing textile. Okay, so for manufacturing siya ng textiles that is used to give a newly worn-in appearance doon sa inyong fabrics. Okay. So, it increases softness and flexibility of your stiff and rigid fabric such as your denim. So, ginagawa niyo mas malambot, mas kaaya-ayang tingnan yung bagong gawang denim because your denim is actually very rough when it is firstly or when it is produced, no? Okay. So, the cellulases digest some of the cellulose on the surface of the cotton. So, sa surface lang, para maging soft siya. The paper, okay, so another use of your cellulase is the production of paper. Okay, so paper producers use cellulase to complete the breakdown of your wood chips to paper pulp. It forms a better pulp and the paper are produced with smaller bleach, easing environmental pollution because you actually place bleach doon sa paper pulp ninyo para puputi siya, okay? Because a normal paper pulp is usually colored darker than your produced white sheets, no? Pero by using bleach, yan, napapaputi niyo yung final product, but it also harms the environment, okay? Lalo na yung bleach. So with the use of your cellulase, since cellulase is organic, okay, unlike your bleach, kapag napunta siya sa environment, it gives milder, okay, effects. Okay, it lessens your environmental pollution. Next, let's go to consumer goods. So detergents are also added with industrial enzymes to clean under relatively mild conditions and to remove spots not accessible to soaps. Okay? So proteases, these are enzymes that digest your proteins. Okay? It digests the most difficult clothing stains which contain proteins such as your grass, blood, and sweat. So usually class kasi, what composes your dirt, okay? Yung mga stains na napupunta sa damit, usually ang components nila are either lipids or protein. So, dapat meron kang enzymes present in your soap or detergent that digest lipids and proteins. Okay? So, soaps with lipids, okay, remove greasy stains. So, dito class siguro lipase ito. Soaps with lipase or the enzyme that digests your lipids remove greasy stains because your greasy stains contains lipids. Okay, so amylases are also added to digest starchy residues galing sa mga pagkain. Okay, so yung karamihan sa atin kapag kumakain masyadong excited, so tumatalsik sa damit. So yan, kailangan yung ginamit mo if food usually yung napupunta sa damit. You have to use a detergent or a soap that contains your amylase, okay? So that it will digest the starch present in the food, such as your oatmeal, the chocolate, and mashed potatoes. These enzymes are effective at mild condition and save water heating bills. Bakit water heating bills? Kasi yung iba kasi para matanggal yung yung stain pinapakuluan pa. So, 'di ba, magastos pa siya. By just using detergent, you will not there's no need for increasing the temperature to remove the stain. So, mas mabilis, mas mura. Okay, medical uses. Because of their specificity, enzymes are used in medicine. Okay, so bakit? Because your medicine dapat ita-target niya lang yung cells that needs, okay? The active ingredient. So dapat specific. So enzymes are used, 'di ba? Because they are highly specific. So they are used in some pharmaceuticals to attack certain types or target certain types of cells. So several enzymes are used to dissolve blood clots in patients with such disease as your lung embolism. So this is, ito yung tinatawag mo kapag nagkaroon ng clot inside the lung, stroke when your clot is present inside the brain, and heart attack when the clot is found inside the heart. So these proteases, plasminogen, or you call them as plasminogen activators. When you say plasminogen activators, these are proteases that catalyze activation of plasmin. Okay, so you have your plasmin. It must be your plasmin is an important factor in fibrinolysis. Okay, fibrinolysis. So when you say fibrinolysis, ito yung pagbe-breakdown ng fibrin polymers. Diba? Yung fibrin ninyo, yun yung nag-cause ng clotting. Okay? So, in order for you to break down this fibrin polymers, okay, you will form during blood clotting, you will have to use plasminogen activators. Okay? Your plasminogen will digest or break down the fibrins or the polymers of fibrins in order to reduce the clot. Okay? So, your proteases are specific for protein plasminogen, which is converted to plasmin and degrades the blood clot in 30 to 60 minutes. So, napakabilis, diba? So, if usually sa mga patients na nakakaramdam ng chest or heart or chest pain, yan, pinapainom sa kanila yung medicines that contain your proteases that specifically attacks the plasminogens, okay, or the fibrin clot. Okay, so let's go to enzyme active site. Okay, so small portion of your, okay, nakalimutan ko palang ibanggit yung biostoning, okay? So your biostoning class dito saan yun ba? Yung sa denim, okay? Your biostoning, ato. Okay. Biostoning, this is more to make your denim cloth more uniform, your uniformly aged look. Okay? Mas parang kumbaga parang same lang din sila ng function nitong stone washing. Okay? So, your biostoning, this relies on the action of enzymes. Okay, to modify your fabric surface. So, yung surface lang din ng denim cloth, minamodify of the biostoning process. Okay, so this allows your denim to produce a more softer surface, okay, and worn out look. Yan kasi yung ina-achieve ng denims, di ba? Yung parang gamit siya pero bago siya. Okay, so yan, uniform age look yung binibigay ng biostoning process to your denim or freshly produced denim cloth. Okay, so let's go to enzyme active site. So a small portion of your enzyme structure where catalysis happens is called your active site. So dito sa active site, nagbaband yung inyong substrate. Okay? So, this is a crevice or pocket. Okay? So, palalim siya or parang maliit na butas on the enzyme structure that represents only 1 to 5% of the total surface area. So, 5% lang siya to 100, no? Or over 100, ganun lang siya kaliit doon sa site ng inyong, doon sa structure ng isang enzyme. But its function is very important because dito nangyayari yung connection or dito pumapasok yung structure ng inyong the structure of your substrate. So mamaya may picture. I will show you the image. For example, the enzyme hexokinase, which catalyzes the breakdown or pinapabilis niya, it increases the rate of reaction of the breakdown of your glucose to provide metabolic energy. So usually ATP itong binibigay or ginagawa or the production of ATP is what your hexokinase catalyzes with the use of glucose. So notice that the active site is located in a crevice, okay, sa palalim na portion. So the glucose enters the site and is bound. The enzyme must change shape before the reaction takes place. Although the reaction happens at a small active site, the enzyme's three-dimensional structure is very important. So look at this. Ito yung inyong hexokinase. Yan siya. Okay? May maliit na butas sa gitna. Okay? And ito naman ang glucose. Okay? So, after reaction or your glucose, okay, the substrate binds with your, okay, it goes to the small crevice found at the center of your hexokinase. And it will be, ayan, it will be bounded there, binding the glucose. So, yan ang itsura niya, class. So, in letter A, the enzyme hexokinase and its substrate, okay, glucose. Ang color dapat yung glucose is pink, okay? Hindi lang siya naklaro because black and white photocopy lang kasi yung ginamit ko dyan na, okay, na-copy. So, before binding, okay, in letter B, after binding, note the top half part of the hexokinase closes down on top of the glucose as predicted by the induced fit model. Yan. So, kailangan, when you say induced fit model, mababanggit yan mamaya, this just shows that after your substrate binds with your enzyme, okay, the enzyme changes or slowly changes its structure. It closes down para hindi na matanggal yung substrate or yung pagkakadikit ng substrate. Okay, so, to function effectively, an enzyme must attract and bind the substrate. Okay? So, hindi siya bibitaw, okay, yung enzyme ninyo sa substrate. So, a chemical reaction is catalyzed once the substrate is bound. So, nagkakaroon lang ng increased rate of reaction once your enzymes binds with your substrate. So, kapag walang binding na nangyari between your substrate and your enzyme, wala ring mangyayari na catalysis or increasing of the rate of reaction. Okay? Ayan. So, the process is shown below. You have your enzyme, letter E, and you have your substrate. Okay? When binding happens, you will have a complex enzyme or you have an intermediate called enzyme substrate intermediate, yung E bonded to S. Then, kapag nagkaroon na ng binding between your enzyme and your substrate, magpuproceed na to catalysis. Okay? So, saka ka palang magkakaroon ng catalysis kapag nag-bind na sila. So, after the catalysis, okay, so you will produce your enzyme and your product. Ito yung the letter P or your enzyme now decomposes your substrate into different products or letter P. Ayan, and the regeneration of your enzyme. So, remember class, your enzymes are not consumed. So, therefore, sa pinaka-end na process, nandyan pa rin yung enzyme. Okay, buo pa rin siya together with your product. Okay, so nakaproduce ka ng product but your enzyme is also still present. Okay, so nagkakaroon lang ng regeneration because your enzyme is not consumed in a chemical reaction. For example, the hydrolysis of your maltose by the enzyme maltase. So maltase plus maltose, binding will happen and it will produce the maltase and maltose When your intermediate now or the binding is done, Okay? So, catalysis will occur. So, ano yung kinakatalyze dito na reaction? Hydrolysis. Okay? So, meaning your maltase here is a hydrolase because it catalyzes the hydrolysis reaction of your maltose. Okay? So, the end products, you will have your maltase again, your enzyme, plus the two glucose units or the two products. Okay, the two final products. Okay, so your maltose was broken down into two units of glucose. So the shape of the active site is unique for each different enzyme, which determines what specific substrate can bind. Okay, so enzymes are stereospecific. Okay, they are stereospecific, which means that each enzyme catalyzes reactions for only a limited number of different reactant structures. So, for example, maltase binds to maltose, okay, to glucose joined by alpha 1,4 glycosidic bond, but not to lactose, okay, producing your galactose and glucose. So, yun lang. When you say stereospecificity, ito lang din yung pagiging specific ng enzyme to substrates. Okay? So, the enzyme also distinguishes maltose from cellubiose. Two glucose joined by alpha or beta 1,4 glycosidic bond which differ in the linkage. So, dito sa stereospecificity class, meron parang yung enzyme ninyo, it recognizes the difference in the stereoisomers or yung pagkakaiba-iba ng shape ng structures ng isomers niyo or ng substrates. Okay? So 'di ba pareho namang ayan 1,4 glycosidic bond yung maltose and yung cellubiose. Pero, ang maltose niyo is alpha while your cellubiose is beta. So, kahit ganun, same sila ng structure, nagkakaiba sila, di ba? In their 3D structure. Okay? So, at the alpha and the beta. So, very, matalino yung mga enzymes niyo. Hindi niya, since beta yung cellubiose, hindi niya it will not act upon your cellubiose kasi hindi siya the same with your maltose that has an alpha 1,4 glycosidic bond. Okay? Between two glucose units. Okay, so ganun din sa lactose, 'di ba? Ang lactose ninyo composed by galactose and glucose. So it is not the same with your maltose that is composed of two glucose units. May galactose dito. So hindi rin mag-a-act upon sa lactose yung maltase. So ganyan na yung stereospecificity. Your enzyme recognizes the difference in stereoisomers or enantiomers doon sa structure nila. Okay, sa spatial arrangement ng atoms. Okay, so stereospecificity of enzymes is very important, means by which the cell controls its biochemistry. So the complementary relationship between the active site and the substrate structure is part of the enzyme specificity. So according to the lock and key hypothesis, the substrate acts as a key that fits into the appropriate active site which is the lock. Okay, so ang substrate ninyo, ang susi, ang enzyme naman or the active site of your enzyme naman ang lock. So the active site are not rigid. So remember, unlike 'di ba matigas yung lock natin pero yung active site niyo it is actually flexible but like your lock, the active site bends to a certain degree when the appropriate substrate binds. So it accommodates. Ang tawag dito class is accommodation. Okay? Flexible yung active site so pwede siyang mag-change ng kaunti sa shape kapag nagbabind sa kanya yung appropriate substrate. So, ina-accommodate niya yung shape ng substrate. the induced fit model states that the active site adjusts its structure in order to prepare the substrate enzyme complex for catalysis. So, ang sinasabi, you have two main hypothesis. First is the lock and key hypothesis. Second is the induced fit model. Ayan. So, tandaan nyo yan sila. Okay, yung induced fit model, ang sinasabi niya, nag-change ng kaunti yung shape ng inyong active site found in your enzyme to accommodate the binding of your substrate. On the other hand, yung lock and key hypothesis naman, this says that your active site of the enzyme serves as a lock and a certain type of substrate will enter it. Okay, so yun lang yung key na yun, yung substrate na appropriate sa enzyme ninyo. Siya lang ang makakapag-unlock noong inyong enzyme. Okay, so para magkaiba, remember that lock and key says that your enzyme or the active site is rigid enough to not let other types of substrate attach to your active site. On the other hand, the induced fit model naman, it says that your active site is flexible to accommodate the shape of your substrate. Okay, so this shape is rigid enough to exclude very dissimilar. Okay. Sana tayo. Okay, so the induced model active site prepare the substrate. Okay. It means that the active site is having a flexible shape. This shape is rigid enough to exclude very dissimilar substrate. Ayan. So, kapag ayan na, kay sinamarize na pala. This shape is rigid enough to exclude, or yung active site niyo is rigid enough to exclude very dissimilar substrates. That is the lock and key hypothesis. However, it is also flexible enough to accommodate and allow catalysis of your appropriate substrates. Ayan. So that is the induced fit model. So your lock and key hypothesis and your induced fit model actually helps each other in performing the enzyme specificity. Okay? So nagtutulungan yung dalawang yan para ma-produce yung concept that your enzymes are very specific towards its substrate. So, tama sila both. Okay? Lock and key hypothesis, matigas yung active site para hindi makapag-bond sa kanya yung other types of substrates, but it is also flexible enough para ma-accommodate niya yung binding of your substrate. So, here is the sample. So, enzyme-substrate interaction illustrating both lock and key hypothesis and induced fit model. So, the correct substrate you have here. So, ginamit niyo yung different shapes. So, syempre, ito ang correct substrate mo kasi siya yung mag-fit dito sa shapes found in your enzyme. Okay, fits the active site, lock and key, diba? This substrate also causes an enzyme conformation change or the changing of the shape of the enzyme that positions the catalytic group to cleave the appropriate bond. Okay, so this is the induced fit model. So, ang substrate, you have the square and the circle. So, this is the key, this is the lock. Oh, so, yan. So, ito ang, that shows your lock and key hypothesis. Now, in here, okay, reaction complex, nagbind na yung enzyme mo with the substrate already entered the crevice found in your enzyme. And this is what you call the induced fit model nung nagbind na siya. So, nagkasya siya, diba? Okay, so there's a slight change in the shape of in the conformation of your enzyme. So, napansin nyo, if itong part na to kanina nakataas, nung nagbind ng substrate, okay, it locks, it forms a lock doon sa structure para hindi, or para maghiwalay, okay, yung para hindi, or to bind your substrate inside the enzyme. And then after that, after the catalysis, ayan, you will now produce, nahati na yung substrate ninyo to produce the products, the square and the circle. Ayan, so your enzyme now releases the products na magkahiwalay na. So that is the whole function of your enzyme. So the flexibility of enzyme explains enzyme binding specificity and how the enzyme converts reactants into products. An enzyme is a dynamic catalyst. So, bakit siya dynamic? Nagbabago siya ng shape. Okay? The enzyme shape and the reactants both begin to change as the enzyme attracts the substrate into the active site. The alteration of an enzyme shape helps the changing of the reactant to the product. Ayan. So the alterations are, so pwedeng bonds are being broken or bonds can be formed, charges can be moved, your positive and negative, and new molecular substituents added or removed. An example of an actual metabolic reaction, let's have the enzyme hexokinase. So sikat yung hexokinase, no? Although siya ginagamit na sample. So again, your hexokinase catalyzes the transformation of glucose to glucose 6-phosphate. Like many enzymes, it requires a second reactant, okay, your adenosine 5-triphosphate or your ATP, which supplies the phosphate group that is transferred to the glucose. In addition, you also have your metal ion or your Mg2+ is required as an activator in this reaction. So the enzyme can start the catalytic process with all the needed requirements. Okay, so dito meron kang metal part, your cofactor, you also have your ATP, and you also have your hexokinase and glucose. Okay, so to get your glucose plus your ATP and your metal ion will produce energy for the body. So when glucose binds, the hexokinase shape changes to bring the ATP close to the carbon 6 of the glucose. So ayan, dito na natin makikita kung paano ba, anong nagiging effect ng pag-change ng shape ng inyong enzyme to produce products. So dito, para mapaglapit niya yung ATP sa carbon 6 ng glucose, your hexokinase changes its shape, forcing the transfer of phosphate to this specific carbon. Kasi kung hindi siya mag-change ng shape class, hindi magkakalapit yung ATP and carbon 6 ninyo. So, hindi malilipat yung phosphate group galing sa ATP to the carbon 6 of the glucose. So, hexokinase makes this reaction fast in several ways. First, proximity catalysis happens such that the enzyme acts to bring the reactants close together. So, ang tawag dyan, doon sa pinalapit niya yung the action of making your ATP and carbon 6 closer to each other is what you call the proximity catalysis, which is the action of your hexokinase. Okay, so second, the action ng hexokinase positions the reactants so the proper bonds will break and form. So, ang tawag mo naman dyan is productive binding hypothesis. Okay, so it gives the position, okay, pinoposition niya yung mga bonds found in your glucose and ATP in a way na madali siyang mabe-break and may mga bonds na mapo-form. Okay, so dalawa ang action, okay, na binanggit dito. Always remember, the close or the proximity catalysis and the productive binding hypothesis. So the reactants are bound oriented in such a way that products result. Glucose reacts with ATP 10 billion times faster than it would be in the absence of enzyme hexokinase. So, bakit kailangan mo ng mabilisan? Hey, why do you need hexokinase? Kung pwede namang hayaan mo lang yan sila, ma'am. You just let them be yung ATP and glucose. Diba? So, remember, the production of energy between your ATP and your glucose is highly needed because your body or each cell found in your body uses energy in order for it to function and develop and grow. Now, if mahina yung process ng energy production mo, lantang gulay ka na kinabukasan. Okay? So, your hexokinase is very important in the body. It is a very important type of enzyme. So, successful catalyst siya. And this large increase in the reaction rate is essential to life. Cellular reactions are too slow to keep cells alive without enzymes. So, completely new enzymes are designed by biotechnologies with their understanding of enzymes. So, gumagawa, ito na yung artificial, ito na yung tinatawag natin na artificial enzymes produced inside the laboratory. One example niya is your antibodies, specifically your immunoglobulin proteins. So, they are produced that bind tightly to a molecule. The strong attractive forces strain the reactants. Okay. Ayan. So, the reactant molecule will change its shape to fit the binding site. This mode of catalysis is what you call the strain hypothesis. Kasi sinistrain mo yung substrate, yung shape ng substrate in order for it to fit doon sa inyong antibodies or your enzyme. Okay, so wherein the antibodies act as enzyme, which is very important in the natural enzymes and these artificial antibody enzymes. So, they serve many important industrial and medical applications. So, tandaan dito, yung ginamit ninyo or the enzyme that is artificially produced is the antibodies or the immunoglobulin protein. So, here is the action. Ayan. Palitin natin siya. Kalaki. Ayan. Ayan. So, here is your enzymatic site. And here is your substrate. Now, what happens in the strain hypothesis? Straining, coming from the word strain. Okay? So, kapag, diba, hindi siya yung perfect, it does not perfectly fit doon sa shape ng active site. Now, ginagawa niya, it enters the active site and it changes the substrate, changes its shape para lang ma-fit siya doon sa active site ng enzyme ninyo. And now, it will produce a new shape or a newly shaped product. So, yan yung tinatawag natin na strain hypothesis. Okay? The substrate is being forced towards or inside your enzyme or your active site in order to produce a new type of shape product. Okay. Next, temperature and pH effect of enzyme catalysis. So, gaano na ba ito kahaba? 1 hour. Okay, so temperature and pH effects of your enzyme catalysis. Malapit na ba tayo? Layo pa. Okay, so the enzyme's catalytic structure is affected if there is any change in protein structure. Okay, so alam nyo na yan, class. I already explained that. Its activity will be lost if an enzyme is denatured. Okay, so dahil nawawala yung 3D structure ng enzyme when there is denaturation. So the treatments that cause the decrease or stop an enzyme catalyzed rate of reaction are strong acids and bases, organic solvents, mechanical actions, and high temperature. So remember that some of the amino acids that make up enzymes have side chains whose charge depends on the pH. So, ang structure ng ating amino acids, they have positive and they have functional groups, okay? Or yung R, diba? So, yung R doon or the functional groups have different charges na nagde-depend sa pH of the solution. It means that enzyme catalysis is affected even a slight change in the pH. So, like for example, side chains can either be neutral or negatively charged with the carboxylic acid. So, di ba ang carboxylic acid natin is COOH. So, when it has the charge, COO, negative. Okay? and either neutral or positively charged with those having amino group. So, ang amino group natin always positive, diba? NH2 or NH3 plus. So, as the pH changes, the enzyme charge specifically located at the active site of the enzyme also change. So, ang main goal or the main lesson here is that when pH changes, naiiba niya yung charges, okay? Or naiiba niya yung charges in the amino acids found on the active site of your enzyme. So, the enzyme catalysis is affected, okay? When your pH changes, the charges of your enzyme also changes, and it affects your catalysis reaction or the speeding up of the rate of reaction. So there's an optimal pH for each enzyme. So tandaan, lahat ng enzymes may optimal pH. When you say optimal pH, this is the pH in which your enzyme acts effectively or most effectively. Okay? Ayan. So the change is, okay, here you will see rate of reaction. Okay, for example, ito, yan. Ang optimal pH ng inyong enzyme is about the range of 6 to 8, okay, pH. So, more than that, 'di ba? Okay, less than 6 ang pH mo, wala kang na-perform na products. And then, sa gitna niya, okay, saan may for example, 7 yung optimal pH. So, sa 7 din, may pinakamataas na rate of reaction at may pinakamaraming products na na-produce. Na kapag lumampas ka naman sa range na 8, okay, so 0. So, yan yung example na optimal pH. Your body have developed elaborate mechanisms to control the amount of acid and base present in cellular fluids. So, ano yung mga organs that are included in or responsible for maintaining the pH of the body? You have the kidneys and the lungs. So, they share the responsibility for maintaining the blood acid and base level. So, ito, yan, I hope that this is already discussed sa anatomy and physiology. Okay, included na yan doon. Especially the lungs, bago lang kayo nag-respiratory system, you know how the lungs control the level of pH in the blood. So, if the blood pH shifts outside the narrow range, 6.8 to 7.8, death often results. The body's metabolic processes cannot maintain life partly because of the pH-induced change in enzyme activities. So, enzymes activities are particularly temperature sensitive. That is why our body temperature is also carefully controlled. So, katulad ganun din class. Katulad sa pH, meron ding optimal temperature ang inyong enzyme. So, yun lang yung sinasabi dito. Okay, so at very low temperature, there are few reactant molecules with enough energy to overcome the activation energy barrier, which results to very slow reaction. Kapag malamig, di ba, when you increase the heat or the temperature, mas nagkakaroon ng collision and therefore mas tumataas ang energy, and with enough energy, you can overcome the energy barrier. Kapag malamig, then syempre mahina ang metabolic reactions. Kasi hindi sila masyadong nagko-collide yung molecules. So more reactants have the necessary activation energy as the temperature increases, resulting to an increase in reaction. Pero kapag sumobra, okay, however, when high temperature causes enzyme denaturation, okay, and the reaction rate starts to decrease. So, kapag sobrang heat naman, so nade-denature na yung inyong enzyme, so sisira ang 3D structure nila. So, okay, so that results to decrease in rate or reaction rate. Consequently, both high and low temperatures cause low reaction rates. And the same with pH, each enzyme has an optimal temperature. So the optimal temperature for many human enzymes is the body temperature about 37 degrees Celsius. Kaya nga 'di ba, ang normal na body temperature ng katawan natin is 37. Kasi diyan nagwo-work most of the enzymes found in our body has the optimal temperature 37 degrees Celsius. So the enzyme activity and effect of the temperature is seen on the graph. Okay. Bakit ayaw niyang bumaba? Ano to? Ano nangyari? Z. Ayun. Okay. Hinabahan ako. Okay. Rate of reaction. You see here. So, the optimal temperature for our body is 37. Okay, kaya sa 37 din may pinakamataas na rate of reaction and mas maraming products ang napoproduce. Outside that range, diba? So 0 hanggang 60, 0 din. So, okay. Ayan. So, chemists have long sought enzymes that have optimal activity at high temperatures and more acidic or basic pH. Conditions that favor industrial reactions such as enzymes can be harvested from microorganisms. So, ang ginagawa kasi, yung mga scientists natin or other chemists, naghahanap sila ng mga micro or enzymes found in different organisms that live in extreme conditions. Okay? So, yan, di ba? At high temperatures and more acidic or basic pH. Kasi, para mas marami silang ranges ng enzymes na magagamit. Kasi most ng enzymes na meron tayo that are known are very sensitive. So, mas magandang makahanap ng mga enzymes from microorganisms that are not or have more or have higher or wider range of optimal pH and temperature para mas matagal yung activity nila, 'di ba? Hindi masyadong mahirap i-maintain yung optimal pH and temperature nila. So, you call these enzymes or itong enzymes na ito nakikita sa mga microorganisms that live in extremely harsh environments. For example, sa volcanoes, 'di ba? Napakainit. Places or microorganisms that are found in places with very low pressure, ganon. So, ayan. So, in very acidic and basic environments, yun. So, yan yung mga tinatawag nating extreme or extremely harsh environments. Yan. So, it means that not all enzymes operate optimally around physiological temperatures such as 37 degrees Celsius and pH or neutral pH which is 7. So, less than 5% pa lang class ang nahanap natin or nadidiscover natin ng mga fungi and bacteria that are being cultured and used. Dito kasi sa mga sa 5% fungal and bacterial species, okay, that are being cultured and used, dyan kinukuha or this is where your other enzymes are being collected or harvested. So until now, scientists travel the world to find new microorganisms and new enzymes which are found in hot springs, deep ocean vents, and tropical rain forests. Okay, so some bacterial enzymes commonly used with detergents are seen in Table A. Okay, each protease has different optimum conditions. Ideal for warm water washes, itong alkalase and sabinase natin. Okay? With about 40 to 60 degrees Celsius optimal or optimum temperature. Kapag gusto mo naman ng, you have sabinase, better optimum pH for detergent powders with a pH value of 8 to 11 pH. So, basic siya. Okay, itong sabinase. It is very effective in basic environment. In comparison, your alkalase is better for liquid soaps, okay, with the average optimum pH of 8. For industrial and institutional laundries, aspirase is frequently used with active temperature of 50 to 70 degrees Celsius and pH up to 12. So, basic nung esperase natin. Okay, so kapag detergent powders kasi, more on basic sila. Okay, so the industries uses Savanase. Kapag liquid soaps, magkaiba ang pH kasi ng liquid and detergent soaps or powders. Kapag liquid, siya, more on the neutral side, so alkalis ang ginagamit. So, ayan ang kanyang graph or table. Okay. So, next, enzyme regulation. Okay, so enzyme regulation, in here, ang dinidiscuss dito is paano nare-regulate yung action ng enzymes. Okay. Ayan. So, enzyme catalysis is under careful cellular control because they are vital to life. So, iniingatan talaga ng cells ninyo yung action ng enzymes because they need it a lot. They use it a lot. A lot of different mechanisms are used by living cells to change the rate of substrate conversion to product. A new group of atoms is covalently connected to the enzyme in the process called covalent modification, while another molecule is non-covalently bound to the enzyme. So the change in protein structure results to a decrease in enzyme activity. You call that as enzyme inhibition. And pwede ring increase in enzyme activity, enzyme activation. So, ito yung first two ways, okay, in regulating the action of your enzyme. Enzyme inhibition and enzyme activation. So, example of this is the cellular control of your hexokinase. Okay, so the hexokinase catalyzed reaction is often not needed when cellular energy levels are high. Kasi yung function ng hexokinase? It catalyzes the conversion of your glucose to energy. Okay? So, kapag mataas naman yung cellular energy level, so hindi mo na kailangan ng additional na conversion ng glucose to energy. So, ang ginagawa dyan, your cells will inhibit, okay, the action of your hexokinase. So, since this enzyme begins the breakdown of glucose to yield cellular energy. So, how will your cells control the action of your hexokinase para mabawasan yung production niya or catalyzation niya ng conversion ng glucose? So, number 1, the glucose 6-phosphate concentration increases if glucose is not needed for cellular energy, and this compound inhibits the enzyme activity by binding to the hexokinase enzyme. Okay, so imbis na ang glucose ang magbabind with your hexokinase enzyme, yung product niya na glucose 6-phosphate. So kapag the glucose 6-phosphate bonds with your hexokinase enzyme, then there will be no energy formed. So this enzyme control is what you call product inhibition. Bakit? Because ang ginamit ng cell ninyo na factor in stopping or preventing the action of your hexokinase enzyme is the product itself, glucose 6-phosphate. Okay. And because the glucose 6-phosphate is a product of the reaction. Hexokinase does not use more glucose than needed and responds to the overall cellular energy state. Okay. There are two other ways. Ito na yung pangalawang way kung paano mo ma-regulate yung action ng enzyme. Okay. Number 1 is the feedback inhibition, and number 2 is the feed-forward activation. So we visualize the different cellular processes as assembly lines having different enzymes at each step from raw material to finished product. So step by step siya yung inyong process ng ng catalyzation. Okay, so the feedback inhibition affects enzymes at the beginning of the molecular assembly. So if straight line, you imagine it, if the cellular process is a straight line, you will find your feedback inhibition action sa starting pa lang, okay, ng process. Okay, so a final product acts as a feedback and inhibits an enzyme from using too many molecular starting material. So your enzyme, syempre, in a cellular process, it is found on the very beginning. Kasi doon siya, it uses the raw materials at the beginning or the reactants to produce product, diba? So, yung final product found at the end, okay, ayan, a final product is being used, okay, to inhibit, kaya feedback, kasi galing sa pinakadulo, the end, okay, it inhibits your enzyme found at the very start. Okay, so feedback, pabaliktad, okay, yung flow. So a final product acts as a feedback and inhibits the enzyme from using too many molecular starting material. So kapag ginamit yung final product found at the end as a feedback, ang ginagawa niya, it prevents your enzyme from using the materials found at the beginning para hindi siya makaproduce pa ng marami pang products, okay? So, parang going back, no? Okay, yung feedback or feedback inhibition mo. So, yun lang tandaan nyo doon. Sa feedback inhibition, it prevents your enzymes from using products from the beginning so that it will not produce more okay, narito na ako. Okay, again, sa feedback inhibition, ang nangyayari lang, your enzyme is being prevented from using materials in the beginning or from using the reactant so that it will not produce another batch of products, okay? So, doon pa lang, nasa stop mo na yung production ng products, okay, by using your feedback inhibition. In comparison naman, your feed-forward activation often controls enzymes at the end of the molecular assembly line. So, dito, nangyayari ang feedback activation sa pinaka-end ng linya. Imagine niyo, doon na naman yung action at the end of your cellular process. So, meaning sa product side. Okay? Kanina, yung inhibition or feedback inhibition, sa reactant side siya. Itong feed-forward activation sa product side. Okay? So, if there is an excess of starting materials, meaning napakaraming reactants, these molecules will feed forward and activate enzymes, which cause the whole process to move faster. Okay? So, mas maraming, dito sa feed-forward activation, mas marami kang product na napuproduce. Okay? Kasi pinaparami niya yung starting materials or yung reactants that are present. Okay, so both of these control mechanisms serve to coordinate enzyme processes within the cell. Okay, so to summarize, feedback inhibition, so ang main goal niya is to stop the production of more products by your enzyme. On the other hand, ang feed-forward activation, it urges your enzyme to produce more products. So, baliktad sila ng action. Okay, so different drug therapies use enzyme control to selectively affect target cells. So, a drug molecule is carefully designed to bind only to one type of enzyme. So, the drug blocks normal catalysis by binding and causes enzyme inhibition. Okay, so ito na yung one example ng pag-use ng enzymes ninyo or the use of the specificity of your enzymes in certain drugs. Okay, so a mental health treatment or in mental health treatments, a drug is introduced to inhibit specific brain processes. So an example of this is your Prozac. and related drugs or they specifically inhibit the reuptake of serotonin, which is a neurotransmitter. More serotonin remains to activate neurons, which will alleviate your mental depression. Okay, so dito gumagamit ka ng enzyme para mas ma-focus yung reuptake ng serotonin lang, okay, na neurotransmitter or hormone. Okay, so ang serotonin, this is what you call your happy hormones. Okay, kapag mas mataas yung level ng serotonin in your body, so yan, mas napapasaya ka niya, 'di ba? So it is used for depressed patients. Okay, binibigay itong drug na ito for depression. Okay? And then, other neurotransmitters are also responsive to treatment by specific enzyme inhibitor drugs. So, when you say enzyme inhibitor drugs, okay, it only allows one specific, okay, or it makes your drugs very specific to the hormones and other target cells that they act upon. So, a marked decrease in blood cholesterol levels is done by statin drugs such as lovastatin, pravastatin, simvastatin, and fluvastatin. Okay, so, and atorvastatin. So, ito sila types ng mga statin drugs, okay, to decrease your blood cholesterol levels. These drugs specifically inhibit a key enzyme in the cholesterol synthesis pathway. So the statins have an impact although the human body contains thousands of different enzymes. Okay, so dito, when you say enzyme inhibitor drugs, okay, so ito dito sa ating blood cholesterol levels, ang statin drugs, okay, hindi niya tayo pinapaproduce ng fibrin para hindi magkaroon ng clot and also para hindi tumaas yung blood cholesterol level. Yan. So, dito so these drugs illustrate that enzyme controls often has major impact on life because of biological processes that depend on enzymes. So, ang tandaan nyo lang dito, class, okay, when it comes to drug, lalong-lalo na itong enzyme inhibitor drugs, these are drugs that makes your enzymes lose their action, okay? Kasi sometimes yung enzymes, hindi lahat ng activity ng enzymes are positive. Okay? Meron din siyang negative effects. So, some of your enzyme inhibitory drugs, they are used to inhibit, okay, the action of your enzymes in order to not produce a specific type of product that will cause harm to the body. So, ito yung example niya for your statin drugs. Okay, further reading, you have aspirin and stroke, a very potent enzyme inhibitor. Okay, so, your aspirin. Ang aspirin, noon pa ito, sikat ito siya na medicine, pero now, hindi na masyado siya ginagamit. Okay, a carefully designed drug will selectively target only one specific enzyme. So, when you say enzyme inhibitor drugs, okay, isang enzyme, hindi lahat ng enzyme, okay, tinatarget niya. Only one type of enzyme. Specificity is the key, and pharmaceutical industries invest about $1 billion per new drug to achieve the desired result. However, nature sometimes provides a very effective drug for little expense. So, one example natin dyan is your aspirin. Mahal kasi kapag when you're producing pharmaceuticals, napakamahal ng process ng pagiging specific ng inyong drug. Okay? So, mahal siyang gawin. But luckily, your aspirin is less expensive. It has been used for centuries as part of the willow bark tea to produce or to reduce fever, aches, and pains. Willow bark contains the active ingredient salicylic acid that is improved by making the acetic acid ester acetyl salicylic acid or aspirin. So, ito yung buong name niya. Ayan, aspirin. This was first widely available in 1900s. So, aspirin potently inhibits one particular enzyme called your cyclooxygenase. This enzyme has an active site in a protein tunnel. Okay? So, a positively charged amino acid is at the tunnel's mouth, and a reactive hydroxyl or your OH, okay, is just inside. So, ang aspirin structure is ideal for blocking this active site. So, ang ginagawa ng aspirin, it blocks, okay? Imbis na substrate, yung magbabind doon sa active site ng inyong cyclooxygenase, ang ginagawa niya, inaagawan niya ng eksena yung substrate, okay? So, siya ang nagbabind doon sa active site ng cyclooxygenase. Okay, so paano yun? First, aspirin's negatively charged carboxylate is attracted to the positively charged at the tunnel entrance. Then, aspirin's aromatic ring squeezes into and blocks the tunnel. Okay, finally, aspirin's acetyl group esterifies the reactive alcohol so that even after salicylic acid leaves, the active site remains inhibited. So, kapag once your aspirin binds with the active site of your cyclooxygenase, so, nai-inhibit niya yung action ng cyclooxygenase enzyme, okay? It also leaves the active site, but, yun nga, parang nade-denature niya na yung, with the use of your, with the use of your alcohol, it denatures your cyclooxygenase. So, hindi na siya magkakaroon ng effectiveness as an enzyme. Okay, so good example of specific aspirin effect is aspirin's protection against heart attack and stroke. Protection is achieved at very low aspirin concentration. So, aspirin's target is the platelet cyclooxygenase. Bakit? Ano ba yung ginagawa ng cyclooxygenase? Your cyclooxygenase helps platelets aggregate. Nagbubuo-buo sila during clot formation. So, when aspirin blocks, okay, so, due to the forming of the clot, okay, pwede kang magkaroon ng heart attack or stroke. So, ang action ng aspirin, it blocks the cyclooxygenase. Okay, so, ang nangyari, fewer clots are formed. Furthermore, because aspirin's inhibition is permanent, okay, so once na dumikit yung aspirin ninyo sa cyclooxygenase, then forever na hindi na active yung cyclooxygenase enzyme. A small daily dose will have a cumulative effect. 1 baby aspirin a day, so when you say baby aspirin, ito yung dosage niya, 81 milligrams of the drug, decreases the risk of heart attack or stroke by 20%. So, patients can be treated for years with little or no side effects. So, ang aspirin is very safe. You can take it for a very long time and it will not give you side effects. Okay, so that ends the video discussion for your chapter number 6, which is enzymes. I know that this is I think very boring siya kasi puro discussion lang siya class, but gawin nyo lang, pakinggan nyo, and then take down notes lang kayo. Ganun na lang. Okay, so hindi siya maiwasan kasi kahit sa face-to-face class, ganyan talaga ang nangyayari kapag puro concepts, but just just read your material. There's no harm in reading your material. Okay, so assume nyo na maraming objective parts ang manggagaling dito sa enzymes. Because your enzymes are mostly, ang topics niya is just ideas, okay? Or concepts. So, marami dyang objectives like identification and such. Okay, so thank you for watching and I hope you are taking down notes. So, bye!